Okay, let's keep going with skeletal muscle physiology. We had talked through some of the concepts in excitation contraction coupling at the end of the first lecture of the semester. So let's just remember where we started from. So we all have brains. This is your brain. Where's your cerebellum? And in your brain, you might have the idea that you want to move. That's a light bulb coming out of an idea turning on. So you will send an action potential down a motor nerve. And that motor nerve will ultimately end up in skeletal muscle. So in this big, huge skeletal muscle that is subdivided into these little bundles that ultimately come down to a single muscle fiber. And a muscle fiber, again, is the individual muscle cell. So this motor nerve that's traveling with this action potential comes down and actually embeds itself into that muscle fiber. And the action potential that travels down the nerve can get transmitted. Let's say AP action potential here can actually get transmitted across and create a end plate potential, or basically transfer that action potential from the nerve onto the surface of the muscle fiber. When that action potential propagates down into those T tubules, it opens up those ryanodine sensitive receptors, and there is this huge release of calcium into the muscle fiber. And calcium is this really important intracellular trigger to cause cause a muscle contraction, because on an individual actin, there are active sites that are covered by that ribbony tropomyosin. So the action potential, the active sites are hidden underneath that ribbon, and the troponin that binds to the tropomyosin has a binding site for calcium. So calcium binds to troponin, it pulls the tropomyosin off of the active site. Now the myosin heads that are sitting here ready and waiting to bind can bind to the active site, and you can have a muscle contraction. That's as far as we got. And now we want to take that one step further and actually think through, how does opening up the active sites through the release of calcium create movement of the muscle, right? The actual contraction of the muscle. And that is described as the cross-bridge cycle. So in the top of the next page of the notes, there is the words that go through and talk through the cross-bridge cycle. But I'm going to kind of give you a schematic or a drawing as I go through it to talk through what happens. So the overarching goal of the cross-bridge cycle, so here is sarcomere. These are your Z discs that have thin filaments or actin attached to the Z disc. And in the center is the thick filaments, the myosin. And the goal of a muscle contraction is to pull the two Z discs together. So when the muscle is contracting, the two Z discs will become closer, because the myosin heads will bind to the thin filaments and change their configuration. Oops, that one's not the right way. And change their configuration to pull the Z disc close to each other. And that would be a skeletal muscle contraction. So the cross-bridge cycle is basically how that process happens. So we have, again, our actin, which is our thin filament. That's the kind of little blue guy here in my drawing. The actin has tropomyosin wrapped around it. And for the purposes of this part of the story, calcium is present, which means that the tropomyosin has been pulled away from the active site to reveal availability of a binding site on this actin for myosin. Now this myosin head, we've mentioned several times, is a really critical player in the movement of the muscle. So we'll draw it a little bit bigger because the myosin is larger than the actin. In the resting position of myosin, or sometimes referred to the low energy position of myosin, the head of that myosin is at a certain conformational position. And the reason it's called low energy or resting is you can imagine if you could think of a myosin head as almost a slingshot, right? And the rubber of the slingshot gets pulled back. When it's pulled back, the elastic of the slingshot has a bunch of stored potential energy, right? If you let go of the slingshot elastic, boing, it's going to fly back and it's going to shoot your rock across the room. Because while it was pulled back or cocked into position, you stretched the elastic properties of that slingshot, and that's stored in the elastic, ready to be released as the elastic would go back to its resting position. So when the slingshot is just sitting there, not being pulled back by the shooter, the elastic of the slingshot doesn't have any stored energy to it. It's in its resting position. So this would be the description of the resting position of myosin. People take offense to the name resting because it's hard to say truly where the myosin is when the muscle is at rest. So because of that, it's also called the low energy configuration. And I could think of that as when the slingshot is not cocked. So in this position, you can find ATP bound to that myosin head. And it's bound right where you need it. For those of you who are less familiar with cellular energy mechanics, and I apologize, I didn't realize my email was on, it shouldn't ding again. If you're less familiar with energy mechanics, ATP, or adenosine triphosphate, is the main energy currency of the cell. When you talk about needing energy in a cell, you're talking about needing ATP. ATP is a molecule that stores energy, and you can actually break off one of those phosphates. So triphosphate stands for 3 phosphates. If you break off one of that phosphates, it will release energy that can be used by the cell for cellular work. So in the resting position, ATP can be bound, but you haven't yet used any energy. So in your notes, it says the first step is to activate the myosin head. So what it means by that, in your notes, by saying activate the myosin head, if we redraw this, and for the rest of this drawing now, I will just draw the actin without the tropomyosin and the troponin for simplicity's sake. Here is the actin, and then we'll re-add in the myosin here. In step 2, we're going to activate. So here's when it was in resting. And you can, what would be a good color? Let's turn it green for when it's activated. You can activate the myosin head and actually bend it into a different conformational shift, sometimes referred to as the cocked position or the high energy position, by breaking off one of those phosphates from the ATP. So now you'll have ADP and a freed phosphate, and the energy released from this ATP that had been bound from releasing one of the ATPs, that energy is actually used to cock the myosin head. And it pulls the head into a high energy position. And that high energy position should make you think of a slingshot that has just been pulled back. Now, what's really important at this step, this first activation of the myosin head, is that we actually use energy at this step. This step is where we see hydrolysis of ATP. So the energy of muscle contraction is actually energy used to activate the myosin head just prior to the muscle contraction. And we'll go back to that because that's kind of a concept that gets a little sticky for students. We have used energy to take the myosin head from its resting position into this high energy configuration. We used ATP to pull our slingshot back so that we can have that stored energy for whenever we want to release it. So what happens next? We now have a myosin in a high energy state. we have, if you remember from the top of our picture up here, we've got calcium available, so the active sites are available. At this point, the myosin head is going to bind. So here again is my actin. And if we wanted, we could add that binding site from the very top picture. We have our myosin here, and the head is in its high energy state, and now it binds. It still has that ADP and that little free phosphate kind of attached on, ready to go. Now this is the really exciting part. We've kind of done all the background work for the magic to happen. As soon as the myosin head binds the active site of the actin, two things happen simultaneously. One, it's going to release the ADP, and the myosin head is going to snap back into its resting position. So let's draw that into our picture here. Oops, still have the orange going. So we've got our actin, and I'm actually going to slide it over because the actin will have moved from this step. Here's our myosin. It is now back to its resting configuration, right? It moved this way from the above picture. The ADP and the phosphate have drifted off. Goodbye. See you later. Thanks for helping out with the process. And the movement of the myosin head from that cocked resting position, hanging on to actin, which goes along for the ride, is called the power stroke. The return to the resting low energy position is what actually moves the actin along. So we have just seen at one individual binding site the cross bridge cycle. The fact that we needed calcium to be available, so the active site was available in the first place. That we needed ATP, or the energy currency of the cell, to be present so that we can cock the head into the high energy configuration. The conversion from the resting position to the high energy position of myosin is the step that actually uses the energy. It's where we use the ATP up in this process. But once that head is cocked and you've pulled the slingshot back, it is ready to just snap back into position. And it will do that as soon as the myosin head binds to the active site, the ATP and the phosphate are released, and it snaps back into its position. And now you can see that this whole actin molecule has actually slid right over in this direction. And we've shortened the muscle with this one step. Now at this point, myosin would stay bound to the actin. If we still had ATP available and another ATP bound, if it jumped on here, it would cause myosin to release the actin, and you would actually start right back up from the beginning. We are back into the resting position with ATP bound, which can be used to cock the myosin head again. And if the actin is still open for business, that new myosin head would bind, the ADP would float off, the myosin head would return to its low energy state, and we've shortened the muscle even further. So this process goes around and around and around as long as two things are there. One, calcium is still available and bound to troponin, which is necessary to keep that active site available on the actin. And two, you have ATP to continue to bind. If there is no ATP available, the myosin head will not release the actin. And that's something that's kind of well known in the general population as what happens when a person dies and they get rigor mortis. Any myosin that's bound to actin at the time of death, if you can't make new ATP to release it, it will hold, and your muscles become incredibly stiff because of this reason. So the binding of actin to myosin, the holding of those two molecules together, does not require energy. The movement of actin and myosin creating a contraction requires energy because of the process required to cock the myosin head. That is the step that uses the ATP. In the PowerPoint slide, I have the same process in cartoon form if you had a hard time following my drawing version of this. And when it comes to the CrossFit cycle, you kind of have to pick a spot to start. So they start in the same place where they have a myosin head in its low energy configuration and ATP is bound, but there is no binding between actin and myosin. The myosin head has an enzyme called myosin ATPase. And myosin ATPase is the enzyme that hydrolyzes the ATP to rip off one of the phosphates, to release the energy, and provide the energy necessary to get that myosin head into its high energy configuration. This is the step that actually uses up ATP. It's the energy required to pull the slingshot back. Now that the myosin head is no longer bound to ATP because the ATP was used to convert it to its high energy state, the myosin head can bind to actin, which causes the ADP and the phosphate to leave. That opens up a binding site that would be accepting ATP if there's going to be more available in the future. As soon as myosin binds to that active site of ATP and the ADP and phosphate dissociate away, we have the power stroke where we actually slide the filament along and the myosin head has moved the actin and shortened the distance between your two Z lines to create a muscle contraction. If there is ATP available, ATP will bind, myosin will release from actin, and around you go to have a continually shortening of the skeletal muscle. So this has described the molecular process by which we get a contraction. In kinesiology, you guys talked about the properties of muscle that contribute to the generation of muscle force. And this is a really important concept to kind of fold in and sew into the fabric of the story that we're making in this class. So depending on your confidence here, it's really important to be able to relate these basic kinesiological principles into the exercise prescription and into skeletal muscle physiology. So let's first give yourself a quiz to see how confident you are about this. Before you even go on to the next slide or before we go ahead in the video, would you be able to answer the question, at what length is there the greatest amount of passive tension in the muscle? When is the greatest amount of passive tension in the muscle generated? The second question is, at what length is the greatest amount of active tension generated? The contractile property that can create force. Is there a muscle length where you'll get a greater amount of force than another? Or is force equal along the entire range of motion? Pause the video if you're willing to engage in some active learning in this format and see if you can write down the answers to those two questions. At what length is the greatest amount of passive force? And at what length is the greatest amount of active force? All right, since you're turning the video back on, or you're ready to get those questions answered, these are two length-tension relationship graphs that you should be very well familiar with from kinesiology. The first is your passive length-tension graph. So basically, as the muscle stretches, so on this end, the muscle is on slack, you are progressively lengthening the muscle as you move along the x-axis. As you stretch the muscle, you are going to elongate both the parallel and series elastic components of the muscle. So just like taking a rubber band, and at this point the rubber band on either side is on slack, there's no tension at all. As you tighten up the rubber band, you're going to start to generate some tension. the spring-like resistance, is the passive tension. When you hit the critical length is the point at which you start to generate tension. It's when the rubber band is a straight line, but you haven't yet started to pull it. From then on out, any further increase in the length is going to create an enormous amount of elastic tension. And you'll see that kind of exponentially increase the total tension in the passive properties as the length increases. So the answer to the question, at what length is there the greatest amount of passive tension, is full length, right? Because you've just taken that rubber band, the farther you can stretch the rubber band, the more tension there's going to be up until the point when you tear it. Active force is more related to this molecular machinery that we just talked about. So I asked, is there a length where you have the greatest amount of active tension? And the correct answer is the mid-length. So when the muscle is very short, it's represented by this kind of A figure in the picture. And really think through. So these little sketches here are the sarcomere. If that image isn't, come back. I was going to change my color. I thought I was allowed to, but maybe not. Oh, here we go. All right, so you have your Z line and your thin filament. And then in the center is your thick myosin with the myosin heads coming off. On the actin, right, you have active sites, numbers of active sites here. The greatest number of binding between myosin head and active sites on the actin will create the greatest amount of strength of the muscle. The amount of force generated by each sarcomere is equal to the total number of cross bridges formed. So when the muscle is so short as an A, you can actually see the actin filaments are overlapping each other. They're covering up their own active sites. So if the total amount of force generated by each individual sarcomere is equal to the total number of cross bridges formed, when the muscle is very short, not all of the active sites can interact with myosin heads. As the muscle begins to lengthen, you can see how you're starting to stretch the actin and you're starting to expose a greater and greater number of active sites on the actin to the myosin heads. At the midpoint of the muscle, there is the greatest number of overlap between myosin heads and active sites on the actin. And since the amount of force generated by each sarcomere is equal to the total number of cross bridges formed, if you have the greatest alignment of myosin heads and active sites on actin, you're going to get the most force. As the muscle continues to lengthen, you are going to start to lose active force again, because as you can see in D, look at all these myosin heads that have no active sites to join. This is getting messy. Let me cover that up so you can see it. All these myosin heads have no active sites to bind to. So you're actually going to get fewer total cross bridges. And that's why we see a decrease in force, active generated force, as the muscle lengthens. So these are really cool, and I love stuff like this about physiology, but we don't really exist in this world, right? We don't have our passive properties separated from our active. So we often describe our muscle force as a total length tension curve. So the two curves that we've already talked about, active force is the green, and you can see that it rises up until the midpoint, and then it will start to decline. The purple is the passive tension. That's our rubber band that just is going to exponentially increase as we get longer. But a muscle that's within a human body has both properties, and the total force generated by the muscle is a combination of the passive tension of the properties being stretched and the active force being generated. So when the muscle is in its shorter length, the active force really dominates and carries the day, right? Because there really isn't passive force at very, very short lengths. Once you get past that critical length, then you really have a combination of passive tension and active force. And the passive tension offsets that decrement in active force. It kind of flattens the curve. Gosh, it feels weird to talk about flattening the curve, but in any other context besides COVID, but that's also what happens here. So that's a really important concept to keep in mind when we're prescribing exercise and trying to maximize muscle force in patients that are really, really weak, that there is alignment of that muscle that might be optimized to create success in an activity or in exercise. Okay, types of muscle contraction. This is another important point that you should already know the answer to this one. So see if you can write down the definitions, and then you can put me on like triple time to get past this part of the video. But if you're not 100% sure about the definitions, then leave me on regular time and listen to them. So isometric activation. Isometric means there's no change in length, right? So the activation of the actin and myosin is used to stabilize joints and body as a whole. You can use an isometric contraction. If you allow a maximal isometric contraction, that would be the summation of the length, tension, active force, and passive tension. And you guys already know from tests and measures that we can't take the muscle out of the human. So we use a maximal isometric contraction as an indicator of a muscle's peak strength, right? That's our position that we take in our tests and measures and our manual muscle testing. A concentric contraction means that the internal torque is greater than the external load and the muscle shortens. So in a concentric contraction, you're creating a greater internal torque than whatever the external load is, which allows the muscle to shorten. Whereas in an eccentric contraction, the external torque exceeds the internal torque. So the nervous system is driving the muscle to contract, but is elongated in response to a more dominant force. I know those are definitions you've heard 1,000 times, so I hope you took the time to just see if you could write out the definitions so that you can appreciate kind of what's already in your brain before this semester started and what do you need to kind of pack in there while you're studying this semester. Oh, my pages all got tangled together.